My year as fellow at the Wissenschaftskolleg, dear rector, dear Barbara Stolberg-Grillinger, a very warm welcome to you, is vivid in my memory, even though I have to confess it was 25 years ago, a quarter of a century ago. Many fellows of this year's cohort in Wallotstraße, a very warm welcome to all the fellows, which most of them I met in the early morning. So many fellows of this year's cohort in Wallotstraße could talk about why such a fellow year is so unforgettable and stands out categorically from the sometimes faster and sometimes slower flow of time. Fellows like Margaret, whom I warmly welcome today. It's a great pleasure to have you here as the keynote speaker of the Ernst Mayr lecture, as well as all the other fellows of her year. A very warm welcome to all, but also to all our guests, friends from Berlin's universities, members of the Academy, students, guests, journalists. A very warm welcome to all of you. I myself remember my year as fellow in Grunewald so strongly because, as a humanities scholar, I came up with completely new ideas in the fellows community, developed new areas of specialization, and entered into new working relationships. The Ernst Mayr lecture in 1989 played a central role in this, even though the keynote speaker at the time was not fellow of our year. There was only his lecture. At that time, Jared Diamond, an evolutionary biologist and geographer from UCL in Los Angeles, spoke on the topic of rich and poor. And most of us will know that this was also the title of a book of him published in 1989 that caused furore, we would say, in the German language. The core message of the Ernst Mayr lecture and the book of the same name is, on the one hand, that the very different natural conditions on the continents, and not differences between the human races, form the basis for the global economic dominance of the Western and the Asian cultures. According to Diamond, the transition to the agricultural societies, which was decisive for human development, was primarily characterized by the availability of domesticable plant and animal species. According to him, Eurasia had an advantageous special position here from the very beginning. And on the other hand, according to him, the superior development and globalization of Eurasian cultures is also due to the geographical orientation of the continents. I skip a lot of other details, but I confess that his considerations during my fellow year, 25 years ago, in the Ernst Mayr lecture made a strong impression on me at this time, and are partially responsible for the fact that I have since paid much greater attention to such geographical connections in my actual academic work. So Mayr lectures are crucial. And I'm quite sure also this Mayr lecture. The Mayr lecture of Diamond, 25 years ago, was only the second in a long and I think proud series of lectures jointly organized by the Wissenschaftskolleg and the Berlin-Brandenburg Academy of Sciences and Humanities. The selection is the responsibility of a group of equal members of permanent fellows. I'm greeting them also, and members of the Academy, whom I would like to thank very, very warmly for their dedicated work in selecting so wonderful speakers every second year. You have to wait a little bit for the next one. The list of names is like a who is who of international evolutionary biology in a broad sense of the word evolutionary biology, from B for Bonhoeffer to Z for Zollikofer. Most recently, Hopi Hoekstra from Harvard University 2 years ago. Ernst Mayr, after whom the series is named, founded it in 1979 with his first lecture on the philosophy of biology, perhaps some remember of. Ernst Mayr, who died in 2005 in the biblical age of 101, was elected an honorary member of the newly constituted Berlin-Brandenburg Academy of Sciences and Humanities, the former Prussian Academy, in 1949. And in a way, Mayr was returning to Berlin, where he had studied at what is now Humboldt University, received his doctorate in a very young age, and worked as a curator at the Zoological Museum of the University of Berlin, today's part of the Naturkundemuseum, Natural History Museum, from 26 to 32. Mayr became an honorary member as the founder of modern evolutionary biology and one of its most important communicators, but also as a world-class ornithologist and historian of the scientific history of evolutionary biology and biology as a whole. And the Mayr lectures since 1979 have documented that he did not stop his scientific work even in an old age, but was active himself and gave good advice to others, such as the two Berlin institutions with the idea to establish such a lecture series. Today, as far as I know and I have learned, we are talking about a completely new field of evolutionary biology, if I may venture this assessment as a complete layman in this field. It is obvious that many animals develop in a microbe-rich aquatic environments. However, it was not until recently that biologists have discovered that associations with microorganisms are critical, as I have learned, and I'm astonished and impressed and curious to hear more of this. Are critical, those associations, for the normal biology of most, if not all, animal species. Once again, we can expect the Ernst Mayr lecturer to make an important, if not central, contribution to the further development of evolutionary biology, in a broad sense, as I said, of the term. But I shouldn't, as an absolute layman, not try to introduce the speaker. And I'm extremely happy that the speaker is now introduced by a far more experienced person, and so I deliver the podium to him, but I deliver the podium with, again, greetings to all of you. It's wonderful to have you here. It's wonderful to be a partner in this Ernst Mayr lecture. A very warm welcome, especially to you, and now the podium for the introduction of the speaker. Please.
Thank you, Mr. President. Thank you all. It's wonderful to see you all here, to see the good turnout. This is about life. It's about the beauty in life. It's about celebrating how diversity, how different forms of life populate and make us interested in biology. How do we know as kids how that works? Right? You might have seen movies by David Attenborough, they would push many people into the awe of what it is, life on Earth, right? He's standing with his white shirt in the middle of, I don't know, somewhere in the forest, and telling us about ants, about birds, about you name it. When did that start? I think the beginning to love life started way at the beginning of humanity. If we talk about academies, think about later, I don't know, Georg Förster, one of those people in European history who went on ships. He joined Captain Cook and went around the world and went to describe people and describe plants, collect them, plant and animals. What would he takes a bird, he takes this animal and describes them, comes back and makes a living, an entire life career on this one trip on a ship as a young man. Charles Darwin really is not that much different. Went on the Beagle, spent 5 years on Galapagos and other places in the world collecting birds, right? Looking at the beaks and understanding what is this diversity, right? And it's no surprise that Ernst Mayr also worked on evolution and was an ornithologist working on birds, collecting the beauty of life, describing it and thinking about where does it become difficult, right? If you take Charles Darwin, The Origin of Species, even the title of the book, The Origin of Species, is as if we would know where is the origin and what is a species. And then Ernst Mayr coming in and saying, well, but look, we don't even know what a species is. What is a species? Right? Where does it stop? Is it just a bunch of individuals that look the same? No, it's not. Right? So Ernst Mayr goes there and spends a life thinking about what is a species? Where does it stop? Where does it end? Right? He proposes the concept of biological species, of the group of animals. He thinks about animals. I mean, in the end, he loves birds, right? So the group of animals that would interbreed, that makes a species, right? Of course, if you then think about evolution, and that comes later, where does a species begin in time, right? Humans, where do humans begin? 100,000 years ago, 5 million years ago, when they stopped interbreeding with something that was somewhat similar or not similar, I don't know. Right? People can talk on conferences about where does a species begin and where does it... but in the end, they all think about the beauty of life. And then comes other biologists and say, if you only think about birds, and maybe humans, you're missing out on the most beautiful part of it, the one that we don't see. I mean, almost all of the cells, the living cells in the world, are microbes. Almost all of the species, whichever that is, right, are microbe species. The diversity of life on Earth started 4 billion years ago. And it's only a tiny part of that diversity that is then multicellular organisms that then develop into birds that Ernst Mayr can study. The diversity of life is microbes. Right? And they are not bad. There's a tiny fraction of them that makes diseases. Right? But most of them live. And they live their beautiful life. And it's only that very recent technologies allow us to categorize them, look at them, study them. Right? But it's also not that these bacteria live their life on Mars and we live our lives on Earth. No, we live this life together. We depend on the microbes. We have these microbes in our gut, we have the microbes on our skin, and we are doing well because we have them. And how do we study them? And where are they so apparent that unless you have this interdependency, right, you need somebody to go out there into the world and study that. Somebody who dedicates a lifelong thinking and working on how to do that. How to find a combination or a situation where these microbes and one of those creatures that we can see and touch, right, say a squid, work together, right? And here is Margaret McFall-Ngai, whom we can listen to tonight, who found this squid, a squid that is better than any sort of, I don't know, plane of the army that flies on the sky and is invisible because they do something that when they fly across the sun and you don't see them because they shine light or something, they make themselves invisible. The squid makes itself invisible. And how does it do that? It uses microbes, right? They work together. And these microbes make light and the squid, but I shouldn't tell you this because Margaret is going to tell you this. And so I'll stop here. And what I'll tell you is that the research that Margaret is doing is absolutely fantastic and at the forefront. And if in 25 years we meet again, I think our view of what nature is will have changed a lot, away from just the birds into a much more diverse, much more complex world. So let me tell you one or two sentences about Margaret, just to, because she's so, then we'll not talk too much about herself, understandably so, because the subject is the interesting part. She is born in California and is a Californian, but spends most of her professional life in Hawaii. Somebody thinks that the choice of the squid was linked to the choice of where to work. And Margaret told me that, in fact, the funders at a certain point came to make an audit, right? Why are you spending all this money on trips on Hawaii? What kind of research is that? Yeah, well, I can go to Hawaii. But no, the squid is living there, and so she has to go to Hawaii to do the research there. And so it's a back and forth between Hawaii, where the squid lives, and the lab in California at Caltech. It is Caltech, right? I have to get my memory right, because also my memory relies on my bacteria working together, right? You never know these days with all these antibiotics out there. So has worked in this lab, has worked in California, in Hawaii, and really started off with a small question about the system of the symbiosis between the bacteria and the squid. It was a small project, but it led to a next question. So it became a new project, which led to the next question, which became a new project, led to the next question. And here, what you'll get tonight is a story about many, many intertwined and questions building on each other. And I stop here because we need the time to listen to you. Thank you. Margaret, the stage is yours.
Thank you so much for the introduction. Is this on? Is this on? Okay, because I wander. So thank you so much for the wonderful introduction, Giovanni, and thank you. I would like to thank very deeply the Berlin-Brandenburg Academy for inviting me, and the Wissenschaftskolleg. It's been a great honor to be at WICO for this fall. I've just had such a wonderful time there, and so I'm very, very, very grateful. And I'm grateful to all of you for coming out to see this lecture this evening. So I think I have to take this part off and put this on. I'm taking you to Hawaii now. as Giovanni said. There we go. So this is our study site in Hawaii. And the animals that you're going to hear about live in the back reefs of the Hawaiian Archipelago along the shore. And so we go out in the evening because they're night active predators, as you'll see. And we go out in the evening and we walk along the shore. And they occur in knee-deep water. And we just have dip nets and we bring them into the lab. And so it's an absolutely beautiful place to work. And we're very excited to have had the opportunity to work there. So I'm going to be talking about co-evolving with our partners and what has happened in the association between the Hawaiian bobtail squid and its luminous bacterial symbionts. So this is not a giant's hand. This is one of my technician's hands. So the adult animals are actually very small. And so they're very good candidates for being part of the laboratory because you can keep 12 females and 8 males and produce 50 to 100,000 juveniles a year. So I'm going to start by saying something about Ernst Mayr. So here's Ernst Mayr, and he was, as mentioned, a tremendous contributor to the field of biology with speciation based on reproductive isolation. In other words, animals that mate together and plants and whatnot, or they belong to a single species. And so his world, though, was plants, animals, and fungi, things you could see. In other words, he focused entirely on that, and in fact, he's known for saying, I leave the microbes to the microbiologists. So now, it so happens that what I'm going to be telling you about really began to happen in very, very recently, in the last 16 years or so. So Ernst Mayr died in 2005, as was mentioned, and in 2006, there was a huge technological breakthrough called next-generation sequencing. And what happened was sequencing of DNA became fast and cheap. And so you could now put animals based not on what they looked like, animals and plants and all sorts of other things, not based on what they looked like, but instead based on their genes. And that's a better readout of relatedness. And so a wide frontier was opened, and what happened was the microbiologists got into it. Lots of microbes can't be cultured in the laboratory. So they started going out into nature, and they went and they did, the oceans and a Terra Oceans project out of Europe, the human microbiome projects in very many countries, and so forth. And it was amazing what happened when this cost went down. And I've given you that $6,000 a megabase, euros a megabase, down to about 0.02 euros per megabase of DNA. So that's a huge change. It's now thought, or you can now get, well, I should say it costs $2.3 billion to sequence the human genome. And now it costs about $1,000. So the technology keeps getting better and better. So it's really an amazing breakthrough. And what has happened is we have learned something that we couldn't have known before. The diversity of life is overwhelmingly microbial, as Giovanni mentioned. And this is the biggest change in our view of the biosphere since Darwin. Okay, it's huge. And so the world is dominantly an invisible biosphere. And so what happened was, I mentioned this, what has happened is this is Ernst Mayr's world here, right? And what has happened, this was the model, this is the way the relatedness of organisms was put together by what you could see or not see. And the small things, the microbes were just relegated to a very small number down here. And in fact, it was thought that there were only 4 phyla within this group, and now there are hundreds of phyla. And all of this group, these groups up here, based on their genes, have gone down to being a very thin slice of a portion of the microbial world. They're very highly related to a particular group of the Asgard archibacteria. And so they fit right in there. And everything else on this tree is microbial. So this group here is now the whole, just about the whole banana. So it's gone from, like I said, 4 phyla to hundreds of phyla. What's interesting is that animals and plants, the numbers of phyla in those groups have not really changed. Because what you see, not a whole lot, it's a few things. But what it is that what you see really does reflect their genes in animals, plants, and fungi. So that's not to say that Ernst Mayr studies small things, because if you instead ask the question of how much the biomass is taken up by each of these things, microbes are tiny and they don't weigh anything and they don't have that much carbon. So plants, this is in gigatons of carbon. And so the plants by far and away are the most abundant organisms. And so Ernst Mayr's world, there's a lot to be said for Ernst Mayr's world. The protists are down here, the bacteria are here, and the archaean viruses are there. So now I'm going to talk to you guys about, as Giovanni said, what I've been spending my lifelong research on. So this is, as I said, the Hawaiian bobtail squid and its bioluminescent partner, Vibrio fischeri. And what I'm going to do today is I'm going to give an introduction to luminescence, because it's a fascinating area, and it's something that most terrestrial biologists never think about. And then I'm going to talk about the bioluminescence of the bobtail squid, and using the squid as a way to study symbiosis. And so, although this was the driving selection pressure on this ability to make luminescence is certainly all about evolution, I'll be talking about using this as a model for symbiosis. And what we're doing, and the reason why we've been funded all of our lives to do this, is because animals and plants evolved in a microbial world. And although we've always thought about what has driven evolution in plants and animals has been temperature and pressure and all these abiotic forces, in fact, the biotic forces have been ignored. And in fact, they're really, really huge, particularly when you consider that all animal body plans, for instance, all animal body plans, which there are somewhere between 30 and 40, evolved in the oceans with a million bacteria per mil, a million bacteria per square centimeter. So that's a lot of biotic pressure that marine animals experience. Marine bacteria have been with animals since the beginning of time, since the beginning of the eukaryotic cell. I mean, microbes have always been around. And so it's very likely that the sorts of things that are associated with microbes are highly conserved over evolutionary time. That shouldn't surprise anybody. And so what we've been looking for are various things in symbiosis that are highly conserved. So what we find in the squid, we might find in humans as well. And then I want to finish up by talking about basic science and why it's so important to applied science. Okay, let's start with bioluminescence. Okay, so here we have two ways to make bioluminescence. So some animals make the chemicals themselves that react to make light, and that's called autogenic luminescence. They make their own chemicals. And so I've just shown the firefly here and a few other things down here. So the other way to do this is the other way, is to make friends with microbes. And so there are several species of microbes that are bioluminescent, And they make alliances with animals. And so here's a group of flashlight fishes, a deep sea anglerfish, and this little guy that I'm going to talk about. It's very interesting that, that this is almost entirely restricted to marine animals. So the luminous bacteria, and almost entirely restricted to animals that have vision. And so those are two things that seem to go together. Okay. So luminescence, one of the things, we're all terrestrial here, and one of the things that's really interesting, and something that people who study terrestrial systems rarely see, except with fireflies and some fungi, is luminescence. It's extremely, extremely common in the marine world. And why is it so common in the expansive marine world? Well, the global oceans, just to say that the marine world is the biggest world, and the ocean covers 71% of the Earth's surface, 97% of the planet's total water, and is the largest ecosystem on Earth. It represents 99% of the planet's biosphere. But in most oceans, there's nowhere to hide. In other words, you're out hanging in space as an animal. Basically, you know, it's not like the terrestrial world. Everything is sucked by gravity, principally on, you know, and things will go transiently, birds and insects and whatnot, but everything is pretty well sucked onto the surface. Whereas in the ocean is very, very deep. And so, sorry you guys, this is in feet. This is, sorry about that, but the meters are over here. And so you see that on average, the seafloor is on average about 4,000 meters. And you have these different zones, and most of the ocean is completely dark. Okay, so I've given some examples here of how animals often hide with camouflage. Camouflaging is matching the environment so the organism can't be seen. And so I'm showing that when you have you're living on a complex substrate, whether or not it's on land or in the sea, the environment is heterogeneous. In other words, it's very complicated usually. And so you've got a lizard here that's living on some leaves, and there's been selection pressure, evolutionary selection pressure, for this lizard to look just like those leaves. And this snake, there's the snake's head, it looks very much like the tree it lives on, and this is the camouflage of the wolf. And then down here, there are some marine animals. This is actually a seahorse. This is its head right there, and it lives on algae. And there's been selection for it to have this very elaborate skin extension so that it looks very much like the algae. But in the marine environment, the light field is homogeneous and very predictable. Very, very predictable. And so how in the world do you hide if you're hanging out there and there's 4,000 meters below you and 1,000 meters above you or something like that? I mean, what do you do? So it's really fun. It's a fun thing because they do nothing like what goes on in the terrestrial world. So I'm showing the three options that marine animals have to camouflage. So if you live in a homogeneous ocean or homogeneous environment, there are three strategies that rarely occur in the terrestrial environment. So I'm showing here, this is obviously a school of fishes. And fishes can be, often fishes in schools are silvery, and that silveriness is used by animals to sort of confuse the predator. And then they occur in these very large schools. And that camouflage has to do with the way they turn and whatnot. It really confuses the visual field of the predator. The other thing is that if you're small, simple, or your tissues are thin, you can be transparent. And this is a very, very, very common thing in the marine environment. You can be swimming along and all of a sudden be stung by a jellyfish and you can't see the jellyfish. You know, it's a very, very common thing to be almost entirely transparent. The third thing is to counter-illuminate, and that's what the little squid that I work on does. And I'll talk about that a little bit. So this is a little guy named Cyclothone, and they're little tiny fish. And they're about 2 centimeters long, and along the basal surface, they have rows of photophores, and this is autogenic luminescence. Now, Cyclothone lives in the zone from about 100 to 1,000 meters, and that's called the twilight zone or the mesopelagic zone. And that 100 to 1,000 meters has a very, very, very predictable light field. And so these guys, there's been evolutionary selection pressure on these guys to develop counter-illumination. They match the downwelling moonlight and starlight so that they don't cast a shadow at all. And so anything looking up from below can't see them so well. So it's super, super common. Cyclothone is a very abundant, it's thought to be the most abundant vertebrate on Earth. There are 1 million billion of them in the mesopelagic zones of the ocean. And most of the fishes and different other animals that live in those zones, like squids and things, they have autogenic luminescence in which they counter-illuminate. And what's fun is on a day-night cycle, of course, there's a little bit of a change in the light field of the mesopelagic zone. And what they do is they migrate along isolumes. In other words, they migrate up and down day to night, matching the light field. So it's a very, very effective camouflage. What I'm showing at the bottom is symbiosis with luminous bacteria. So a lot of the animals, this is something pistoproctid, it's a really weird benthopelagic fish. And it emits ventral luminescence from a bacterial culture that it has inside of its body. And what it does is it renders the tissues associated completely transparent. And it allows, and it filters the light out into the environment. And so that is the other way you can counter-illuminate, is to have symbiotic bacteria associated with you. So let's get into what Euprymna is doing with this. So Euprymna is a night-active predator, as I mentioned when I showed you our study site. And this is what our study site looks like at night. We go out in here and collect. And it counter-illuminates during the evening. So I'm showing a lizardfish, which is their principal predator. And what they do is they come out, and I mentioned that the water's only about that deep. They come out and they hang in that water column waiting for a shrimp to go by or something. And they hang there and they emit ventral luminescence of the same quality, same intensity and angular distribution and color of downwelling light to effectively camouflage against the background light, moonlight or starlight. I'm going to show you the anatomy of the light organ is such that they're able to control it, light emission, to match moonlight and starlight, or say there's no moon. They're more dim. And then when dawn comes, they go and bury in the sand. And they sit in the sand all day until just after dusk, when they come out and then forage again. So, how many times did I do that? Okay, so while bioluminescence is something I've studied, I studied my graduate career, I was studying bioluminescence and how the animal I was working on, it was also a symbiotic association, but it happened to be a fish. And I was studying how the tissues are designed to use the light. Well, I started to study this squid, and it had bacteria associated with it. And these bacteria, Vibrio fisheri, are very culturable in the laboratory. And it occurred to me that this could be a system by which to study symbiosis. And I engaged Ned Ruby to come and work on the bacteriology. I'm like Ernst Mayr, I work on animals. And so we began to study Euprymna as a model system for animal-bacterial symbiosis. And it turned out that there was symbiosis in a lot of places. Nicole Dublier is here. She studies symbiosis in hydrothermal vent animals. And when the three of us were junior in our field, symbiosis was kind of backwater. It was like in hydrothermal vent animals, it was luminous bacteria. There were lots of insects that had symbioses. But it wasn't thought that mammals had symbioses with bacteria. And like I said, that really changed. And so we began to think about this as a possible model. And it turns out that this animal is a very good model for studying symbiosis across the animal kingdom. So what is the power of model for the study of complex characters? Why do biologists use model systems? Well, I'm showing 3 examples of things that are extremely complicated. One is that you go from a fertilized cell to 10 to the 13th cells over about 9 months. Extremely complicated developmental program. The brain is thought to have about 10 to the 14 synapses, and understanding how that works. People are turning to the fruit fly and other things like that to try to understand how brain wiring works. And then the symbiosis is also, as I mentioned, we discovered that it looks like many, if not most animals, have symbiotic associations with microbes that are coevolved. And so this is a very, very complicated situation. In humans, there are thought to be somewhere between 10 to the 13th and 10 to the 14th body cells, and 10 to the 13th and 10 to the 14th bacterial cells. So it's really, really, really complicated. So what is the power of animal models? Well, I'm showing here 1901 to the present, and this was actually the Nobel Prize this year. And all of these Nobel Prizes I'm showing here were done on a series of model systems. The one this year was done on this animal over here. And so these Nobel Prizes are all of the Nobel Prizes, all of them, in developmental biology, in physiology, and medicine. So how many Nobel Prizes have been won studying human research? How many in developmental biology? And the answer is 1. And that was on in vitro fertilization. And so the point is that the Nobel Prizes that were won doing model systems were awarded in the hopes that it would help us understand how human biology works. And they're so simple that, and the idea is that these features are conserved, that it would be something that would shed light on, and obviously they won the Nobel Prize because it did shed light on how a very basic thing happens. So looking at the complexity of the human microbiome, I showed you this over there. This is the tree, 10 to the 13th to 10 to the 14th cells of our microbiome comprise something like hundreds to thousands of species. I'm showing here the number of genes. Like, look at the person next to you. They're 50% non-human by cell number, and by gene number, they're only 1% human. So it's pretty impressive. So, and also remember that the microbes in your gut and on your skin, and they're associated with 9 of the 10 vertebrate organ systems. Remember that they're extremely metabolically active. So 18% of the small molecules in your bloodstream are derived from the microbes, mostly from your gut microbes. So we turn to models in the study of symbiosis. And so what I'm showing here are experimental models. And we're exploiting nature's toolkit. In other words, evolution has done some amazing things that help us learn about what's common across animals. So there are a series of natural symbiotic associations that people study as model systems. And they go from simple consortia. So honeybees, for instance, have what is thought to be, always have worker honeybee always has the same 5 phylotypes or species of bacteria worldwide. Always the same 5. And so studying how a consortium is important. Why those 5? We have lots and they seem to be coevolved, but we can't figure out why. Then there are all these others, and so there are simple consortia, and then there are binary systems, which ours is a binary system in which there's one host and one microbe. There are also engineered vertebrates, such as a zebrafish and a mouse. A shared derived character of all vertebrates is the carriage of a very, very complex consortium, from fish all the way up to mammals. Very, very high consortium, and that correlates with the presence of an adaptive immune system. So, why is the bobtail squid a good model? And why have we been studying this for 35 years? So the specific attribute is naturally binary, like I said. There's one host type, there's one host and one bacterial species associated. And it gives very, very high resolution. You can drop into the conversation and know what they're saying to one another. And like humans, the symbionts reside on the apical surfaces of epithelia. Wherever they occur, they're associated with the apical surfaces of epithelia. One of the things that we really like about this symbiosis is that in most of the time, nutrition is important, so that when you take the microbes away, you not only take the microbes away, but you also take away their nutrition, or you change their nutrition, which is it's a confounding factor. In this case, nutrition is not the principal function, but rather the production of luminescence, which is a very clean thing. You can make a mutant, you can make a delta lux mutant that doesn't make light. The animal won't keep it, but you can make one and it will take it in for a short time. So you can have healthy, non-symbiotic animals, is the point. And you can study strain variation, which is really big, different types of Vibrio fischeri and how they behave in the organ. So I'm just curious, has anybody in the audience seen this movie? It's really funny because this is an American movie, obviously, from the 1980s, so it's a really old thing. It's a fabulous movie. If you ever get a chance to see this movie, it's really fun. So these two guys get together for dinner, and when the movie first starts, you think this guy is crazy and this guy's sane. And at the end of the movie, you think this guy is crazy and this guy's sane. And what it is you've gotten a chance to sit there and listen to the conversation and learn about these two people in great detail and understand what their motivations are and how the whole thing works. So this is like Euprymna, the bobtail squid, and its interaction with Vibrio fischeri. This is the human gut. This is like the Kumbh Mela celebration in India, where 110 million Hindus get together. How do you ever figure out what's going on between any two of these individuals and what might be happening in this situation? It's daunting. It's actually almost impossible. So what we do is we do experiments and we find something and we say to the community studying the human microbiome, look there, look there. And I'll give you an example in a minute. So, like I said, they occur along the apical surfaces of epithelia, and that's either mucociliary. So this is the bronchial epithelium, and this is the intestine of a human. And this is Euprymna, and these are the cilia. Here are the cilia here, and these are the bacteria. And the bacteria are red here, and they're associated with these cilia. It's very, very similar biogeography. And so the issue is, how do they get there and how do they stay there? So basically, what I'm showing here is I'm showing you the series of questions that we ask and the range of questions that we ask. So these guys are what is called horizontally transmitted. That is, they're picked up anew each generation. So humans pick up their microbes anew each generation. They come through the birth canal, and that's the first time they're exposed. And so Euprimna, when it hatches sterile, and then it begins to acquire its symbionts. So it's very much like humans. So there are no microbes present in the embryo. Then there's establishment of the symbiosis, and you have to select the right partner. And that's a whole set of stories. How do they get the right partner? There's development. They come to understand one another and make nice. And then there's stability of the symbiosis. How in the world does it happen that the animal doesn't get rid of the bacteria, nor do the bacteria overgrow the host? So there has to be some kind of very stable alliance going on there. We've also we've studied these three areas in the lab, and also their ecology and evolution. And we're very interested in the differences with pathogenesis. And let me just say here, what we have found with regard to that over the years is that it's not a different language. It's not like French and German. It's the same language but used differently. In other words, it's like an argument versus a civil conversation. And so the same genes, the same proteins and whatnot seem to be involved very often in the control of both beneficial and pathogenic associations. Okay, so I did that. Oh, I know what this was. I made that big because that's what I'm going to talk about. That's the example I'm going to use. I'm going to talk about the stability and the symbiosis. So Giovanni said we've got lots and lots and lots of stories, as you might imagine, on all these topics. But I wanted to focus on one, and that is how do they make this stable association and keep it their whole life? So first I have to tell you what the symbiosis is all about. So here's this casual guy is the host. If you cut them open, like this, you cut their mantle, you'll see that in the middle of their body cavity is this bilobe structure. And if you take one of those lobes, and this is a diagram of what you would see there if you looked at a cross section. So this light organ is a total riff. Is there a German word for riff? It's very much like the eye. It has taken the genes from the eye and made another organ that controls light. And so down here you have a lot of the structures associated with the eye, and in the case of the light organ, it's a derivative of the hindgut ink sac complex. So it uses the ink sac in two ways, as a choroid in the back of your eye and as an iris, to make more or less light come out of the light organ. So that's the ink sac. The reflector, which I'm going to talk about at the end, is here. And it's like the back of a cat's eye, that when you shine light into a cat's eyes, it reflects back at you. That's the tapetum lucidum in the back of a cat's eye. And this guy has a reflector in the back so that stray light coming out of the bacteria, which are carried in these 3 crypts on each side, that light goes, if it goes this way, it bounces back because of the reflector. And then it has lenses there that bend the light out into the environment. So the bacterial symbionts, which are here in the center, are, like I said, along the apical surfaces of epithelia here. It's a really beautiful, beautifully organized. So I'm going to tell you about rhythms. And this is, how many of you know Maya Angelou? Yeah, and she's kind of a famous American. Yeah. And she said, everything in the universe has a rhythm, everything dances. And so I'm going to be talking about the fact that symbiosis has rhythms. And so what are the rhythms in the squid-vibrio symbiosis that ensure stability, and how do they work? So I have to tell you how light is produced, because it's important to how it's controlled. So light-generating bacterial molecules, which for those of you who are chemists, FMN, H2 and a long-chain aldehyde plus oxygen are oxidized to FMN, a long-chain acid, water, and light. So this light production, actually I should say, not just this one, all luminescent reactions from the firefly all the way down to those deep-sea fishes are all oxidation. They always require oxygen. So this is how bacteria do it. So what you see is the diel rhythms of the association actually begin on day one. That animal gets colonized within 3 hours of its symbiont. It's beginning to bring the symbionts, even though the bacteria are only 102 or so cells in a background of 1 million others. The animal is able to pick out its symbiont within 3 hours. And then it starts, they go into the organ and they start to grow, and then each day they continue to grow. The hours following symbiont colonization here, you see this rhythm to light production, relative luminescence. So there are these rhythms beginning at day one. Okay, now they only need, as I mentioned, they're night-active predators, and so they only need the luminescence at night, really. So how in the world do they control symbiont light? Because symbiont light is pretty expensive metabolically to do that light production. So they want to, during the day, they want to have it low, and at night they want to have it as high as possible. So how do they do that? Well, so what I wanted to show you was that, is that the bacteria are in green and the host tissue is in blue and magenta. But I wanted to show you that whereas they look kind of like a flat surface, when you turn it on their side, the bacteria make very, very thin layers within the host, within the light organ. They're very thin layers. So every bacterial cell is in very close association with the host tissue. So that's an important thing. And so this is just to show you, in still life, bacterials occur in thin layers closely associated with host tissue. And you can see that here when you do a TEM. Okay. So the host is very clever at managing the oxygen. And the way it does that is it has a blood protein called hemocyanin, and it's the squid hemoglobin. In other words, it's the molecule that carries oxygen throughout the animal. And so that oxygen is carried on this hemocyanin. It's a copper-based instead of an iron-based like it is in invertebrates. And it coordinates the offloading and onloading of this is managed by this molecule. And so I had a great postdoc in the lab who's now a CNRS in Lyon, Natasha Kramer, who studied how hemocyanin works to change the luminescence. So what you have basically is that hemocyanin is oxygen bound, is not available for luminescence. And so what happens is the oxygen, this hemocyanin is carrying this oxygen, and when the pH is really high, in other words, it's very basic, it doesn't let go of its oxygen. But what happens is, over the evening, because of certain metabolism of the bacteria, the environment becomes very acidic, very, very acidic. And so it goes down and down and down. And then that causes, for those of you who are biologists, that's the Bohr effect. It causes the oxygen to be released from the hemocyanin. So the oxygen is released at low pH, and the bacteria become very bright. And so that's how this works. So I'm showing here that it's left the hemocyanin molecule and now is associating with the bacteria. And that oxygen is very important, obviously, for the luminescence. Okay, so okay, so it turns out that modulation of the oxygen is responsible for about 10% of the difference in oxygen over the day-night cycle. How is the other 90% of the bacterial luminescence controlled? Well, what happens is, as I mentioned earlier, every day at dawn, the squid buries in the sand. And at that time, it gets rid of 90% of its symbionts into the surrounding seawater. So that way, it doesn't have to feed them. They don't need them at that point to that extent. The remaining 5 to 10% left behind grow up to fill the light organ again, so that when they come out at night, they're full of bacteria. But they don't need them. The other sort of incidental thing that this does, and releasing you can see the toothpaste coming out of the light organ here. In an adult animal, they just vent 90% into the surrounding seawater. But that also seeds the environment for the juveniles with Vibrio fischeri. So if you go away from adult populations, there are very fewer and fewer and fewer Vibrio fischeri. So it's very important that the juveniles, which hang about after they hatch, are exposed to the bacteria that have been vented every day by the adults. So what you see each day is on a very profound rhythm. The symbiont population varies. So it goes down at the onset of dawn, and then they grow back up, and then there's a time of very slow growth over much of the day, and then they expel and regrow and expel and regrow, and they do that their whole life. And that's, a very important control on the symbiosis. There are several other symbiosis-driven rhythms that I don't have time to talk about, but I thought I'd mention them. So one of the things that happens at dawn is, for those of you who know anything about pathogenic microbes, they often cause an effacement of the surface microvilli. And that's what happens at dawn each day, is they shed their microvilli. So that's on a daily rhythm. Also, the bacterial symbiont metabolism is on a profound daily rhythm. At first, they eat up these membranes, and that's a pH-neutral metabolism. And I mentioned that the pH goes down and down and down over the day, and that's the result of them. They are then fed chitin, which is fermented, and acid is exported from the cells. And that's what creates the acidic environment. And then the last thing is that there are huge changes in gene expression on a day-night cycle. So just before dawn, the host, there's a huge increase in a whole bunch of genes just before dawn. And just after dawn, there's a huge decrease in gene expression. And this happens every single day from the time they're juveniles. So this transcriptional regulation we published in 2010. And I was a great postdoc in my lab, Andrew Weir, who led this project. And in 2001, there was only one publication on microbe-associated host rhythms, and our paper was that paper. And I kept going to the human microbiome meetings and saying, it's very well known that the gut, every aspect of the gut, from its motility all the way down to its molecular biology, is on a very profound daily rhythm. You guys must look at the microbiome. You have to look at the microbiome. And, they kept saying, go away, you're looking at a squid. Finally, they began to look, because it was it became so compelling. Thinking about when you travel and you get off, you've got jet lag. A lot of times people say you should always eat where people are eating where you are. And now people think that you're re-entraining your microbiome. But so this is one example of where a basic research finding, we were able to say to the community, I had to say it again and again, but say to the community, you should look there. So I'm just giving an example of how, as I mentioned, it began to catch on. Here back in 2012, 2013, there was one other article that had been published, and then there's been a 75-fold increase over 12 years. So it's become a very well-documented and very well-studied system. It's pretty exciting. So I'm going to give my last story, which is about discovery to application. And I feel really honored to be in Germany, because I think Germany respects basic research more than any other country I've ever worked in. And in the United States, we're often asked, why are you doing that kind of thing? And, not only do I feel we as our symbiosis can tell the community something about how symbiosis works, but also basic scientists are very curious people, and they often ask weird questions, and sometimes those weird questions yield some fun application. So exploiting the experiments that nature has done through evolution. So I'm going to go back to the reflector, you guys. I'm going to tell you this. What happens, as I mentioned, is here's this reflector, the bacteria in here, and they make light, and the reflector makes sure that the light goes out where it can be used. So I had a great postdoc in the lab, a woman named Wendy Crooks, who began to look at these platelets that make up this reflector. And she did some TEM, and she was actually a biochemist from University of Michigan. And what she did was she found that the reflector is made up of protein. It was the first protein-based reflector described, and she called it reflectin, which I thought was pretty clever. And anyway, so, we've been working on reflectin as a part of the symbiosis for a long time. It turns out that it's on a day-night cycle. Its production is on a day-night cycle, and it's got a lot of interesting aspects to it. But the people began to use it in applications, and I am very proud to say that I've been giving tissue and whatnot to a very smart engineer down at UC Irvine. And so this is Alon Gorodetsky, and he's a young guy at University of California, Irvine in the engineering department. And he's very interested in reflectins and using reflectins to do various things. They're the most powerful proton conductors in nature. So that's one thing. He's done all kinds of very fun things with that. The other thing is that he was able to take the reflectin gene and figure out how to express it in E. coli and make huge amounts of it. And so his lab has been studying this for a long time now. So what has he done with it? Well, lots of things, but I'm going to give one example here. So he just so clever guy. What he did was he impregnated it into the suits of the military, into the fatigues of the military. And so clothing is in the material itself. So infrared radiation is released from the body, is detected by night vision scopes. And this is what a night vision scope sees if the person doesn't have reflectins. And this is what they see if reflectin is impregnated into the suit. So infrared radiation is blocked by reflectin, camouflaging the body form. So that was an amazing, amazing thing that he did. And he's done a whole bunch of other things with this. And I just wrote him before I came to give this lecture. I wrote him and I said, I just want to let you know that I will be talking about your work in my lecture. And he wrote back and he said, oh, we've got to get together. I've got all kinds of other ideas. And I want to talk to you about it when you get back from Berlin. So I'm excited to get together with him in January and push forward on the sorts of things he wants to do. But the whole point here of me mentioning this is I feel really deeply that pioneering basic research provides the foundation for efficient and effective approaches to the challenges we face. And that's very Max Planckian, as I understand it. Basic research is really important. So now, I'm going to finish up here, and I'm just going to say that I've done all this work with Ned Ruby, and this is us back in 1989, and us in 2024 at a party at Caltech. And we're celebrating 35 years, and we got to celebrate the coming down of the wall, 35-year coming down of the wall. It's quite a different impact, of course, but we're very happy to have been able to work together on this for all this time. It's really been fun. So here we are, and I am deeply embedded in Ernst Mayr's world, as I mentioned. And so I just wanted to show you the babies. So here we have a female squid, and she lays a set of clutches, and that's a clutch of about 300 or 400 eggs. They are 21 days embryonic, and then they hatch like crazy, and the students go crazy. And so this is you know, maybe 100 or 150 babies, and they're 2 millimeters in size. Each one is 2 millimeters. And so they study, much of the lab studies the onset of the symbiosis, how these babies get their symbiont. And so it's a really fun adventure. I'd like to say that since we started, a lot of people have joined us in our quest. And so I'm showing here the current labs with a principal focus on the squid-vibrio system. Because a lot of what's been done in this area has not been done by us, by Ned and me, but rather our academic children, grandchildren, and great-grandchildren. And they're across the United States, and some of them are in Europe. And so it's been a terrific adventure, and it's really important for me to express my great gratitude to all of the students and postdocs and people who have been willing to come and work in the field. And I'm showing the system started at Scripps Oceanography when I was a postdoc. And I am here, actually, there's Ned and me back there. And we're celebrating, every year we get together, as many people who can make it get together in a meeting to talk about where we are with the squid-vibrio system and what's been going on and who's doing what, and trying to get them not to compete, but rather collaborate. And so I'd like to say thank you of them, of course. And then all of the funding agencies that have given us such generous funding over the years so that we've been able to do our work. So with that, is the light still off? So with that, I'm going to say mahalo nui loa. So this is a little guy in the morning. He says, I'm going to go to sleep now. I'm going to sleep now. Aren't they cute? Okay, and with that,